Hi, welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. In this content, I'm going to be showing you a bit of data that's going to be investigating how ketones may have an impact in terms of increasing a particular cancer progression in a cellular environment.
Learn your body, a science-based education.
Now, this particular research comes from a paper that I fully dissected, as well as took notes and have that study linked for you to this content so you can check it out for yourself. But in brief, let's jump into a bit of the methods and then we'll get to the data itself. Essentially, what the researchers did was they took fibroblasts, which are essentially support cells that are found throughout the body but are certainly found in tumors as well. And these fibroblasts were genetically modified to overexpress particular enzymes that are key regulators for the production of ketones. So these fibroblasts will then presumably be able to produce far more ketones than they normally would in a cancer environment. And then what they did really simply is that they co-cultured them. So that means that they added these fibroblasts, these genetically modified fibroblasts, along with cancer cells, a specific breast cancer line. So only one breast cancer line known as MC7. So this MCF7 line is then put on a plate along with the fibroblasts, and then they did a series of experiments off of that. So what did they find? Well, one of the clues that they found is that with the overexpression of these ketone enzymes in these fibroblasts, when they're co-cultured with the cancer cells, the breast cancer cells, you can see that there's higher levels of mitochondria that are located in the cancer cells as a result. Now that's by comparison to a control fibroblast that does not overexpress ketones. So if you have the comparison there, the red is the indication of mitochondria. So you can see there's clearly more red in the two conditions where the overexpression occurred. However, of course, you can't necessarily jump to the conclusion that just because you have greater mitochondria that you have more cancer cells. Although that is certainly an interesting proxy that does tell you that most likely you're going to get more cellular energy out of those cells. In most cases, that is. So the researchers then investigated the actual number of cells, again in the co-culture, and they did, in fact, find that the cancer cells were far, far more numerous when they were co-cultured with fibroblasts that had that genetic manipulation to then express these ketone-producing enzymes. So another piece of evidence that these fibroblasts that produce these ketones are having an impact on the cancer cells and their proliferation, their division. Next, the researchers looked at tumors after a number of weeks of allowing these cells to grow, and they found that the tumor mass, the tumor size, was significantly larger and had more weight if they were co-cultured again with fibroblasts that produce these ketone synthesizing enzymes. And finally, they did an experiment where they removed the fibroblasts, so that's not part of this experiment. They only injected the cancer cells, but they genetically manipulated the cancer cells to express one of two enzymes. And these two enzymes are key enzymes that allow the cancer cells to break down the ketones for cellular energy. And what they found is that only with one of the overexpressions, the ACAT enzyme, you see an increase in tumor metastasis, in cancer metastasis, which implies that ACAT is most likely the rate limiting step, or at least one of the rate limiting steps when it comes to the use of the ketones. So if you have more ACAT, you essentially have a bigger gate that allows more ketones into the cell and allows for that conversion to cellular energy. So ultimately, what can we take away from this paper? Can we say that ketones are going to increase the proliferation, the growth of these cancer cells? Well, actually, not really, because the researchers actually never measured the ketones in the media in the liquid that was surrounding the cancer cells. So we don't actually know if there was an increase in ketone production. Now, if I had to guess, most likely yes, that was the case. There was an increase in ketone production, but they really should have added that in there. That is a huge, huge factor, and that could certainly completely change the overall conclusion of the study if they did measure it and they found that ketone differences were, well, there were no differences. However, most likely there were differences. So based on, with that assumption, which is a dangerous thing to do, but with that assumption, we can say that the environment wherein fibroblasts can produce more ketones and support this particular cancer cell line, and that is a key point here, this particular cancer cell line, this breast cancer cell line. That does not mean that this would apply to all other cancers, just to the one studied here, that leads to greater proliferation. And with that said, hopefully you found it informative, and I hope I have the pleasure of speaking with you in the next one. Have a good one, guys. Bye.